A very good morning, everyone. Myself, Ankita Sharma, and I'm working as a postdoc fellow at Department of Animal Biosciences in University of Guelph, Canada. So today, I have the pleasure of sharing results for one of our study that is a part of the project for to understand the stress resiliency in small ruminants. So today, I'm going to present my talk on topic screening ovine circulatory microRNAs as biomarkers of microbial stress. As all of us are well aware of fluctuating climate conditions that leads to increase in environmental stresses, including heat stress, and due to increase in environmental temperature, there is a subsequent increase on microbial load, which further increases the high rate of infection in animals and raising concern for animal health and welfare. Among microbes, their E. coli is one of the commonly presented pathogen in environment and produces lipopolysaccharide, which is a potent PAMP and has been associated with numerous bacterial pathologies. For example, mastitis, enteromastitis, and toxemia, sepsis, colitis, acidosis, and leaky gut. So all these pathologies causes huge production and reproduction losses, reducing the meat quality and also increasing the susceptibility in animals. So when the microbial infection happens, it initiates the crosstalk between the neuroendocrine and immune system. Like for example, an LPS, it activates the HPA, that is hypothalamus pituitary adrenal axis, that further releases the stress marker cortisol and further stimulate the immune cells to release cytokine to induce the immune responses. So these interactions have been highly influenced by the epigenetic mechanisms, for example, microRNA, whose participation has been highlighted recently in various inflammatory diseases, and that tends to regulate gene expression which are involved in important inflammatory pathways. So what are these microRNA? So these microRNAs are small 22 nucleotide base pair long non-coding RNA that regulate the gene expression post-transcriptionally. And what are circulatory microRNA? These are emerging as a new class of biomarkers which are released in biofluids as a signaling molecule for cell-to-cell communication and have been recently identified to be regulating the immune cell and the immune cell development and responses, which are a crucial part to understand the pathogenesis of inflammatory diseases. There are certain positive aspects of these circulatory microRNAs, such as they are quantifiable, they are very sensitive, and they are remarkably stable in biofluid, which present themselves as a non-invasive sensitive diagnostic biomarker for various diseases. Well, microRNA have been well characterized in human, mice, and number of numerous diseases and cancers, like including cancers, but there is very less information available on the small ruminant, and especially in ovines. And these gap led us to design the objective of our study, that is to screen and identify ovine circulatory microRNA regulating LPS-induced immune response. And to achieve this objective, we designed our study in two parts or two steps, as assessment of circulatory biomarker in LPS challenge lamb as a step one. For that, we selected 18 lambs of 80 to 90 days old from a pathogen-free environment, and LPS dose was given at 400 nanogram per kilogram dosage via intravenous route. The fever response was assessed by measuring the hourly rectal temperature, and blood was collected at every 2 hours as 0 hour, 2 hour, 4 hour, and 6 hour. The serum was isolated and assessed for cortisol measurement, cytokines levels using multiplex ELISA with the panel of 15 pro and anti-inflammatory and chemokines. Also, we selected few mRNA which have been associated with LPS in past in literature, and for that we selected 7 different microRNAs. The step two, was followed by the step one. After having the preliminary information on microRNAs, we decided to go for large-scale expression profiling and functional analysis of circulatory microRNA. And for that, we selected the array-based approach, which contains 84 different microRNAs specific to ovines and along with 12 controls. So for this, we choose QIAGEN, a commercially available PCR array that is from QIAGEN, specific for the ovines. So the quality analysis and the differential expression analysis was done using QIAGEN Gene Globe Analysis Center, and differential expression was analyzed in T4 as a post-challenge stage compared to the T0 as a control or pre-challenge stage. So after identifying the differentially expressed microRNAs, the functional analysis was performed. So first step was to identify the target genes that have been involved, have been targeted by these microRNA. And this was done by target scan, MIRDB, and Miranda tools. After having the genes, the target genes, the gene enrichment analysis and pathway analysis was performed by WebG Salt Server and COBAS. Further, the network analysis was performed between the microRNA and mRNA relationship, and the cluster analysis was performed to identify the microRNAs which are located closely in the genome. Let's take a look at the results. So first of all, we noticed that there is an immediate increase in rectal temperature that started right after post-LPS induction. Within 1 hour, it increases and it remained high until 3 and 4 hours and dropped down by 6 hours. And it came back to normal by T7 and T8, which indicated the feverish response occurred in the lambs post-LPS. Similarly, the level of cortisol shot up at 2 hours with a peak response and remained high at T4 compared to the basal level at T0 and come back to normal at T6 by T6. So similar kind of response was noticed in cytokines level. As we can see, IL-6, interferon gamma, and TNF-alpha as a pro-inflammatory cytokines, they all increase at T2 and T4 and drop down at T6. It dropped down at T6, and there was no significant differences between T2 and T4. Both of the time points have high response compared to the basal level. Similarly, the anti-inflammatory marker, the IL-10, was also increased at T2 and T4. It was slightly increased more at T4 compared to T0 and dropped down at T6. However, there is no significant differences was noted in T2 and T4 time points. Similarly, the chemokines, they also increased after the LPS challenge, and they were high at T2 and T4, and coming back to started dropping down by T6, and they were normal by T8. The data is not presented here at T8. So as we can see, CCL2, CCL3, CCL4, CXCL10, they are increased post-challenge LPS challenge stages. However, among 15 cytokines, 2 cytokines, IL-4 and IL-1 beta, did not change at any time point, whether it is pre and post-LPS stages. And also the other 3 cytokines as IL-1 alpha, IL-2, and IL-17 did not express at any pre and post-challenge time points. Looking at the microRNA that was selected based on the literature with their association with LPS. So microRNA 2 to 3, 1 to 46, 200B, 29B, and 145, all of these were increased significantly at T4 compared to the T0 time point. And interestingly, all these microRNA regulate signaling molecules of DLR4 pathway, which is the PAM recognition for LPS. And the signaling molecules that were controlled by these microRNA are TRAF6, malprotein, MYD88, and IKK alpha. Also, the microRNA 223 and 1246 they have direct effect on increasing the IL-6 and chemokines level. Additionally, microRNA 223 is known as the positive regulator of proliferation and differentiation of neutrophils, and 1246 tends to increase the TNF-alpha levels. So the other two microRNA, as 132 and 31, microRNA 31, they were downregulated at T4 compared to the T0 stage. And these, the downregulation of these microRNA were also associated with the induction of immune response. Like for example, microRNA 31 targets the FOXP3 gene, and downregulation of its own expression leads to increase in expression of FOXP3, which in turn increases the development of the T regulatory cells, which participates in immune tolerance ability. So after having this preliminary information, the array was performed, and here we can take a look at the volcano plot. As you can see, the red dot shows the increase in fold change in these microRNA, and the green dot shows the downregulation of expression of these microRNA, and the black dot indicate that there is no change in expression of these microRNAs. So in number, we profiled 84 microRNA, and out of which 6 did not express at any time point, and 28 no change in expression, and also 34 microRNA were noticed as upregulated with more than twofold change expression, and among these 9 were significantly upregulated, and the downregulated were the 8 microRNA, and all of these were significantly downregulated. So here are the list of differentially expressed microRNA, and if we take a look at the upregulated microRNA, the first microRNA as 369.3V was the highest increased microRNA with the fold change of more than 140 times, and followed by 495 3P microRNA with almost 58 fold change increase, and then 376 A3P with 32 fold change, and so on. And if we take a look at the downregulated microRNA, all these microRNA have a comparable fold change with almost 2 to 2.5 fold downregulation in their expression as compared to the T0. Interestingly, all these microRNAs were found to be presented on the ovine chromosome 18, which highlighted the importance of chromosome 18 in regulation of immune responses. And also, this information led us to our next step, to establish the relationship between the differentially expressed microRNAs. And for that, cluster analysis was performed using the information available in the MIR-based database, and MetaMIR-Cluster program was employed to identify the clusters where differentially expressed microRNA are closely located on the chromosome. So in cluster analysis, 4 different clusters were identified, with 2 major clusters having differentially expressed microRNA more than 2 presented in a cluster, and the 2 were the small. So if we look at the cluster number 1 as 379 by 495 cluster, this total 7 differentially microRNA were found to be located within this cluster, and the total length of this cluster was only 11,000 base pair. The difference between the location of one microRNA to the other was less than 3 kilobase pair on the genome. Similarly, if we look at the second cluster as 376-487, 3 differentially expressed microRNA were noticed here, with a total length of 12,000 base pair. The third one is a very small cluster of only 2,000 base pair, have 2 microRNA identified in this cluster, and the fourth one is the 6,000 base pair with 382 and 453 cluster with 2 microRNAs in there. The importance of identifying these clusters is that they can transcribe as a single polycistronic primary transcript, which can have a collective effect on the other microRNA and the other target genes. That called as a collective effect of the whole cluster on the downstream processing of the pathways. So after having the differentially expressed microRNA, we identified the target genes. And for that, three different tools were employed as MIRDB, Miranda, and Target Scan. A very stringent criteria was applied to identify the target genes, and only those genes which are commonly identified by at least two softwares have been used. So a total of 1,966 target genes were identified, and these genes were subjected for the functional and gene enrichment analysis using the GSTAL server, which identified the important transcription regulation biological processes. Let's take a look. So here, if you see in the biological processes, most of the gene belong to the biological regulation, metabolic processes, response to stimulus. And if we see here in the cellular component, it's more membrane and protein containing complexes. If you look at the molecular function, it is more protein binding and nucleic acid binding activity. So, important gene ontology terms were identified that were affected by these 15 upregulated microRNAs as regulation of gene expression, regulation of RNA metabolic processes, regulation of cellular biosynthesis, and circulatory system development. In molecular function, it's more in the regulatory activity of transcription and DNA binding activity, and the cellular component is more in the neuron and the plasma membrane part. So after identifying the important gene ontologies terms, we try to do the pathway analysis using the COBAS, and the identified differentially expressed microRNA, we found that they participate in acute phase response while influencing mostly the signaling pathways. As here you can see, the most enriched pathway that were affected by these microRNAs are PI3K AKT signaling, immune system, MAP kinase signaling, RAS signaling, FOX signaling, and signal transduction, which reflects the immune stressed state post LPS challenge in lambs. So after having these pathway analysis, we try to have the relationship between mRNA and mRNA as a network. So in a network, a big network was identified, and that comprises of 16 differentially expressed microRNA, and comprising of 2,689 genes were identified. So, and if we take a look at the subnetwork, so these are the four important microRNAs: 200B, 29B1, 495, and microRNA 223. That lies commonly and shared genes which are commonly targeted by these microRNA. So, and if you take a look at the microRNA 223, so these are the genes that are associated with the immune response as TNF alpha, lysozyme, LPL, IL-6, IGF-1R, HMGB1, and so these are important immune regulatory genes or immune induction genes, which is regulated by microRNA 223. So it means that it could be used as a stress biomarker for various studies in livestock species. So in conclusion, first of all, the monophasic fever response was elicited post-challenge in post-challenge lambs was noticed. A balance of immune response was determined by increased pro and anti-inflammatory cytokine levels. Among the total 91 microRNA, 51 for differentially expressed, and among those, 41 were upregulated, with 15 significantly upregulated, and 10 microRNA were found to be downregulated significantly. 4 microRNA clusters were identified that were all presented on chromosome 18. So that led us to our next step to explore more about the chromosome 18 in its role in immune processes and its regulation. With identification of 1,966 genes from these upregulated microRNAs, several important pathways were identified, which indicates that these microRNA play important role in induction and regulation of immune responses. So overall, a set of circulatory biomarkers, that is ovine microRNA, regulating the acute phase response were identified, and these could serve as a biomarker for livestock in future studies. And with this, I would like to acknowledge my supervisor, Dr. Neil Caro, and my lab mates and friends, and our funding partners. First of all, my Tex Elevate, as it thanks for providing me as a postdoc fellowship, and the second is the Food from Thought funding program from UOG and Sephora of Excellence Fund. For providing the funding for this project, and Ontario Ministry of Agriculture and Rural Affairs for letting us to use their amazing pathogen-free environment research station for sheep. Thank you, and I would like to take any questions if you have any. Thanks.